Now I am delighted to introduce Dr. Jill Koshiol, who is a senior investigator in the Infections and Immunoepidemiology branch of the National Cancer Institute. And she will be speaking us today about circulating immunological markers in hepatocellular carcinoma.
Thank you so much for the introduction, Betsy, and really to all of the organizers for inviting me, and even more importantly, just for putting together this opportunity for all of us to talk and share about the work that we've been doing on hepatitis-related hepatocellular carcinoma. I'm really happy that I have the opportunity to share some of the work that we specifically have been doing on circulating immunological markers. And just to give you a little bit of background, hepatocellular carcinoma, or HCC, is strongly tied to inflammation. Hepatitis B virus and hepatitis C virus are two very major risk factors, very important risk factors for the development of this cancer. And both of them have impacts on inflammation and immune response. A few previous prospective studies have found associations between HCC and circulating soluble ICAM1, IGF-BP3, and IL-6. These previous studies have had limited or even no ability to try to understand how these markers relate to the development of cancer in chronically HBV infected or HCV infected individuals. So another limitation of these studies is that they tended to look at just a small number of proteins. So we felt that a more comprehensive evaluation of circulating immunologic proteins, specifically in the context of HBV and HCV, would be really useful. And fortunately, we had the opportunity to do that in two existing cohorts in Taiwan, the REVEAL HBV and the REVEAL HCV study. Just to give you a sense of the overall design of REVEAL, from 1991 through 1992, individuals from 7 townships in Taiwan were invited to participate in a community-based cohort study on virus-related cancers. Over 23,000 were enrolled, and of those, 4,155 were positive for HBV surface antigen, which is an indication of chronic HBV infection. And then of those, 3,653 were HCV seronegative. Of the nearly 20,000 who were HBV surface antigen seronegative, 948 were anti-HCV seropositive. So this got us down to our cohorts that we evaluated in this study. We proposed to extend the field beyond what had been done in previous studies by looking at a larger number of markers in REVEAL HBV and HCV, with the primary aims to evaluate the associations of hepatocellular carcinoma and immunologic markers in the context of chronic HBV infection and the context of chronic HCV infection, and then to compare how the patterns of inflammation markers were similar or different in HBV and HCV related hepatocellular carcinoma. And today I'm really going to focus on this third aim, associations that were identified both in the HBV and the HCV cohort. For this study, we used the Olink technology, which has a number of different panels, and we focused on the inflammation panel and the cardiometabolic panel. We chose the cardiometabolic panel because many of the proteins on this panel are actually produced by the liver. So we thought that could be particularly relevant for the development of hepatocellular carcinoma. Both of these panels include 92 markers. And we also, when we did the testing, we included blinded QC samples from 12 HBV infected and 11 HCV infected controls, which allowed us to evaluate how well these markers performed. And based on that analysis, we ended up dropping 82 of the markers, either because they were below the lower limit of detection in the majority of samples, or they had low intra-class correlation coefficients, or high coefficients of variation. And that left us with a total of 102 markers for our analysis. For our primary analysis, we used polytomist logistic regression to compare hepatocellular carcinoma to non-cirrhotic controls and to cirrhotic controls. We ran separate models for HBV carriers and HCV carriers. Now, for right now in this talk, I'm going to focus mainly on the comparison between HCC and the non-cirrhotic controls, but I will come back to comparisons to cirrhosis just a little bit later on in the talk. Here you can see the adjustment factors for these models, and I want you to kind of keep these in mind because, again, a little bit later in the talk, I'm going to talk about some receiver operating curves based solely on these clinical and sociodemographic covariates compared to what happens when we add in immunologic markers. So just tuck that away in the back of your head. For the 3 a priori markers that I mentioned, we used a significance value of 0.05 to determine statistical significance. And for everything else, we used a false discovery rate of 0.1. So getting to the good stuff. Here you can see the odds ratios and the 95% confidence intervals for our 3 a priori markers, plus 4 novel markers that we saw were associated with HCC, both in the REVEAL HBV and the REVEAL HCV cohort. And here in this figure, REVEAL HBV is indicated by the black triangles and REVEAL HCV by the white circles. Just to remind you, an odds ratio of 1 indicates that there's no association between the immunologic marker and HCC. An odds ratio above 1 indicates an increased risk of HCC, and odds ratios below 1 indicate a decreased risk of HCC. And as you can see, of the a priori markers, only ICAM1 was associated with HCC in both the REVEAL HBV and the REVEAL HCV cohorts. Although the other 2 markers did sort of go in the same direction, the expected direction, with IGF-BP3 tending to have inverse associations and IL-6 tending to have positive associations. Of the novel markers, HGF and CXCL11 were positively associated with HCC, and SCF and CFHR5 were inversely associated with HCC. Then focusing on the markers that were associated with HCC, both in REVEAL HBV and in REVEAL HCV. Here you can see the associations for people who had at least 10 years of follow-up. We wanted to get a sense of how long-term these associations were. And as you can see here, the associations with hepatocellular carcinoma typically remained even after we restricted the analysis to people with at least 10 years of follow-up, indicating that they do reflect long-term effects. And we think that that means that in the future, these markers potentially could be useful for risk prediction or early detections in populations with a high risk of HCC. But replication is really important, and we're going to come back to that in just a minute. These findings also have biologic plausibility, since the positively associated markers promote cell proliferation, survival, and angiogenesis, while the negatively associated markers have anti-inflammatory and anti-tumorigenic effects. And again, the fact that the associations remained even when we restricted to people who had at least 10 years of follow-up, so cancer didn't develop for at least 10 years, suggests that these really are long-term effects that can help us predict who's at risk of developing cancer. An important limitation of this study, though, was that we didn't have any information on underlying liver fibrosis. And advanced liver fibrosis can look a lot like cirrhosis, which might lead to the question, are these associations that we see with immunologic markers really only reflecting an association with underlying liver disease? To address that limitation, we wanted to assess whether circulating immunologic markers were associated with hepatocellular carcinoma among people who already had fibrosis and cirrhosis. Now, if we saw associations with cancer in people who already had fibrosis and cirrhosis, then it really does reflect an actual association with cancer. To address this question, we turned to the hepatitis C antiviral long-term treatment against cirrhosis, or HALT-C study. This is work that my postdoctoral fellow, Ilona Argirian, is leading. And many of these slides are hers, so I definitely want to thank her for the use of these slides. And this is a clinical trial that evaluated the efficacy of maintenance therapy with pegylated interferon alpha-2. And it found that actually this maintenance therapy didn't affect the risk of progression of fibrosis and developing HCC. They didn't see differences between the treatment arm and the control arm. Just to give you a couple of important points about the trial, it recruited people from 10 different sites in the US from the year 2000 through 2004, and included people who had previously failed therapy, who had advanced fibrosis according to liver biopsy, who had no history of hepatic decompensation, and absence of other exclusion criteria, such as liver disease, other than hepatitis C itself. And one really valuable thing, among many valuable things in this study, is that fibrosis was assessed through liver biopsy, which has really been considered a gold standard. For this study, we included 94 individuals who developed hepatocellular carcinoma and matched 2-to-1 controls on those individuals. And again, we used the Olink inflammation and cardiometabolic panels. We actually ended up adding a third panel because it included IL-27, which Jianmin Yuan from the University of Pittsburgh presented at AACR a couple of years ago as having strong associations with hepatocellular carcinoma. So we wanted to be able to look at that as well. For this analysis, we used conditional logistic regression for individual immune markers and their association with hepatocellular carcinoma versus people who were chronically infected with HCV but did not develop hepatocellular carcinoma. And that was our primary analysis. But as something of a secondary analysis, we also took a look at the association between these markers and cirrhosis versus fibrosis among the controls who did not develop hepatocellular carcinoma. You can see the adjustment variables here. And again, we used 0.05 for a priori markers and a false discovery rate of less than 0.1 for everything else. Here you can see the odds ratios and the 95% confidence intervals for the a priori markers. And here I'm considering a priori those that had been found in previous studies, plus the reveal cohorts that I just presented to you, and then novel markers that were identified through this study. And what you see is that among the a priori markers, ICAM, HGF, and SCF were all associated with hepatocellular carcinoma, even among these individuals who had advanced fibrosis and cirrhosis. And that suggests that these associations with hepatocellular carcinoma and what we observed previously in REVEAL and in other studies were not due solely to underlying disease, but instead that they reflect a true risk of developing cancer. In addition to these a priori markers, we identified 7 novel markers associated with increased risk of HCC among people who already had advanced fibrosis and cirrhosis. Restricting to people who had at least 4.5 years of follow-up, again to try to get a sense of how long-term are these effects, we found that the magnitude of the association was generally as strong or even stronger among these people with at least 4.5 years of follow-up. And again, this suggests that these associations really do reflect long-term effects. Now, to touch briefly on our secondary analysis of cirrhosis versus fibrosis among controls, we found that levels of HGF and 5 of the novel markers were higher in controls who had cirrhosis compared to those who had fibrosis, suggesting that high levels of these markers are associated with more severe underlying liver disease, as well as with increasing the risk of hepatocellular carcinoma. Now, here I'm going to jump back to the REVEAL cohorts. I promised you that we'd come back to cirrhosis, and I wanted to dig in a little bit and look more carefully at hepatocellular carcinoma compared to cirrhosis. And here you can see among HBV-infected individuals, if you recall, our primary analysis was using polytomous logistic regression. And when we used that, we actually found that there weren't any individual immunologic markers that were statistically significantly associated with hepatocellular carcinoma compared to cirrhosis. But we also used another approach called canonical discriminant analysis, or CDA, where we could compare the baseline plasma levels among people who developed cirrhosis versus those who developed hepatocellular carcinoma versus those who developed cirrhosis. And this approach allows us to look at all of the markers together and identify which combination of these markers does the best job of distinguishing between cancer versus cirrhosis. When we took this approach, we found that there were 11 markers that differentiated individuals with hepatocellular carcinoma from individuals with cirrhosis. We then plotted the receiver operating curves, or ROC curves, for the model that included only those demographic and clinical variables that I showed you earlier. And then a separate curve for those covariates, plus these 11 markers identified through canonical discriminant analysis. The area under the curve was 0.87 for the clinical and demographic covariates only, compared to 0.89 for the covariates plus the 11 markers identified through canonical discriminant analysis. But these curves weren't statistically significantly different from each other. And you can see that in this figure. The curves are pretty much overlapping. So in this particular case, we didn't see that these inflammation markers or circulating immunologic markers helped us differentiate people who would develop cirrhosis, cancer, versus those who would develop only cirrhosis. Things did look a little bit better for discriminating chronic HBV carriers who developed cirrhosis compared to those who did not develop cirrhosis. Here, polytomous logistic regression identified 6 markers that were associated with the development of cirrhosis, and canonical discriminant analysis found 9 markers that differentiated individuals who developed cirrhosis from those who did not. The ROC curve from the clinical demographic model produced an area under the curve of 0.63, and adding the 6 markers from the polytomous logistic regression models increased that AUC to 0.69. Whereas using the 9 marker model from the canonical discriminant analysis plus the covariates yielded an area under the curve of 0.79. And in fact, that area under the curve using the 9 biomarkers was statistically significantly higher compared to the clinical demographic model only. Now, moving back to REVEAL HCV. So we just looked at chronically HBV infected individuals. Now let's look at chronically HCV infected individuals. Here, when we compared cancer to cirrhosis, the polytomous logistic regression identified 4 markers associated with hepatocellular carcinoma versus cirrhosis. Canonical discriminant analysis found 14 markers that differentiated cancer from cirrhosis. And the ROC from the clinical demographic model produced an area under the curve of 0.73, which increased to 0.85 when we added the 4 markers from the polytomous models, and increased further to 0.95 when we instead used the 14 markers from the canonical discriminant analysis. And again, this model using the 14 markers was significantly higher than what you observed for clinical and demographic variables only. Comparing chronic HCV carriers who developed cirrhosis compared to those who did not, polytomous logistic regression identified 4 markers associated with cirrhosis, and canonical discriminant analysis identified 14 markers. The ROC curve for the clinical demographic model produced an area under the curve of 0.56. Adding the 4 markers from the polytomous model yielded an AUC of 0.75, and the 12 marker model, including the covariates, increased that AUC to 0.88. And again, that AUC for the 12 marker model was statistically significantly higher than that you get from the clinical demographic model only. And these findings suggest that these kinds of immunologic markers might help with the risk prediction, at least among individuals who have hepatitis C virus infection and cirrhosis. And the HALT-C study provides a replication for the REVEAL HCV study, but we still don't really have a replication for HBV. And that's kind of what we've been really thinking about now. How do we try to replicate those findings? One way that we've been thinking about trying to do that is by combining data from 3 different cohorts. The Shanghai Women's Health Study, which enrolled 75,000 women aged 40 to 70 from 1997 through 2000. The Shanghai Men's Health Study, which enrolled 61,000 men aged 40 to 74 from 2002 to 2006, and the Zhongshan City Cohort Study. And the challenge with Shanghai Women's and Shanghai Men's is that these cohorts don't currently have data on hepatitis B infection available. So to use these cohorts, we would need to test all of the hepatocellular carcinoma cases and quite a number of controls in order to find enough individuals with chronic HBV infection. And that's a lot of time and energy, and these are really precious samples. So we've been trying to think about what else can we do to make it worth this effort to do this kind of testing. And one thing that we've been considering is that in addition to allowing us to validate our hepatitis or reveal HBV results, we can also look at the prevalence of hepatitis D virus, which is a really important topic that hasn't received a lot of press and we think is valuable to really dig into. And this is work that Tom O'Brien in my group has been leading. So by doing this testing, we could look at HDV prevalence in Shanghai and in China. And then we could also evaluate the associations between immunologic markers and HDV infection. But all of that said, honestly, I'd really love to hear from you all who are part of this webinar. Are there other ways that it would be valuable to you and to the scientific community to have HBV data from Shanghai Women's and Shanghai Men's? So I'm hoping we can have a little conversation about that at some point, or please feel free to follow up with me. Just to wrap things up, identifying factors that affect the risk of developing hepatocellular carcinoma could have important implications for patients with chronic hepatitis B or hepatitis C infection. And factors that affect the risk of hepatocellular carcinoma might offer important targets for prevention and treatment. And just as one example, HGF is already a target for hepatocellular carcinoma treatment, and it's being considered as a marker of liver fibrosis. So continued research into the novel associations that we observed might help identify additional opportunities for early detection and treatment. And with that, I will stop and take questions.
Thank you very much. That was wonderful. Does anyone from the floor have any particular questions? If not, in the chat, a question that came on early on was have you had a chance to evaluate the effects of Chinese medicine on any of these markers?
That's a really great question. And the answer is no. That's not something that we have dug into, but I am honestly increasingly becoming aware of traditional medicine and how it can have an impact on inflammatory markers. So that's a valuable future focus of research.
Okay, thank you. And another question that came in was did you see any differences between the time that samples were taken, if they were closer to one another versus a longer term or longer time separation between the samples? Did you see any differences?
So the analyses that I presented today were actually all samples that were taken from baseline or as close to baseline as we could get. We did actually do some testing of serial samples just to try to get a sense of how sort of stable these markers are, and it was not a large number of samples. So it's sort of hard to draw statistical inferences from what we saw. But what's kind of interesting is that many of the markers, when we had samples that were taken, say, a year from baseline, and then after that second sample, maybe 2 or 3 years later, there was certainly more variation in samples that were taken 2 or 3 years later versus those that were taken just 1 year apart. Things tended to look fairly similar over time for the cancer cases, but there was a little bit more variation among controls. And that, to me, kind of makes sense because the process that lead to the development of cancer is highly tied to inflammation. So you might expect to see higher levels in general, whereas among controls, somebody could have a cold or have something else that influences these markers and might cause a little bit more variability. Is that getting at the question?
Yes, I believe so. And then I had a follow-up question to that. Have you noticed anything or taken statistics or in the history and physicals of your cohorts, any evidence of any other co-infections that are going on at the same time? I'm thinking particularly of HIV and perhaps an AIDS-associated malignancy like Kaposi sarcoma, because I know their levels of interleukin-6 are quite high.
Yeah, it's a good question. So in these particular cohorts, I believe for HALT C that individuals with HIV were excluded.
Excluded, okay.
And for the Taiwanese cohorts, there's just not a very high prevalence of HIV, so it's not so much of an issue there. Something that we haven't looked at that I also don't know as much of a issue there would be like Salmonella typhi infection or something like that.
Well, certainly, yes. And then the last question that came in was, were the alpha fetoprotein levels measured at the beginning of your analysis?
Yes.
Okay. And those are all the questions that came in, I believe. Anyway, well, thank you again. Great talk. And it's nice to know what's going on in other parts of the NCI, and always enjoy hearing what is going on. So thank you so much, Dr. Koshiol.